analysis of data. And we want to think about why is data important and how does this apply to our experiments. And when we first think about experiments, you really want to think about what is the point of an experiment, okay? And so the point of the experiment, why are we experimenting? Why are we doing all this research and working through this, is taking the time to perform these? And the main reason here should be to gain knowledge, and for us, most of the time, that's going to be gaining knowledge on different conditions. For example, our isopods, our roly-polies, in wet versus dry conditions, okay? But then what are we trying to do with that? What are we doing with that knowledge? You're not just trying to collect a bunch of data or get a bunch of information, but you will want to be able to apply that knowledge to a specific problem. Okay? So when you're thinking of an experiment, the important part isn't just collecting that data, but it's being able to answer a question, to solve a problem, or to be able to apply that to a different situation. And when we're thinking of experiments, in the past, what you've had a lot of are what's known as cookbook labs, okay? And now what we're trying to move or change to are what are known as inquiry labs, which is what you guys will be attempting, okay? With a cookbook lab, the results or the outcome is known. Thinking back to Bio 1, we all did a diffusion lab. Some of us used potatoes, some of us used eggs. And in that lab, if you put the potato or the egg into a cup of salt water, left it overnight, came back and measured it, collected some data from it, the egg or potato would have lost mass, would have weighed less than it did originally before we put it in there. But we knew that that was going to happen, okay? That was the idea that we were losing water. And so that result was known. You knew, and even if you didn't specifically know, then your teacher knew what was supposed to happen, okay? Whereas in an inquiry experiment, your results are unknown or unexpected, and they vary, okay? So over here, there was an expectation of what would happen or what should happen. But in an inquiry experiment or inquiry lab, it's unknown and it usually varies, okay? Looking at this quote from the College Board, they're talking about these types of labs. They're talking about these types of experiments. And so the focus of the labs has shifted from student achieving predetermined results to students making independent observations, posing their own questions, and determining how to investigate these questions. And that's the big thing. How are you going to investigate these questions? You're going to do this by collecting data. Because remember, our main focus here is to explore and introduce ourselves to how we use mathematical analysis of data. And when we're collecting data, we want to collect lots of data. The more, the better, okay? So, what type of data can we collect from our experiment? Before we actually talk about the different types of data, we're going to explore, we're going to kind of backtrack and remind ourselves about what's variables, what factors are in an experiment. So one of the first variables that we want to remind ourselves of is of the independent variable. And remember that the independent variable is the factor that is changing or the condition that's changing. And that could be something like temperature, okay, from winter to fall. Or the temperature that we set ourselves, like in a hot house. Then you also have your dependent variable. Remember that these are important factors in an experiment. Your dependent variable is what you measure as or of the results of your independent variable. Okay? These conditions produce this result, and that's what you're measuring. So this could be something as simple as the number of isopods, roly-polies, okay, in a specific environment because of this temperature. And then the last little reminder that we need is of our control group. So you might have observed ahead of time isopods in their natural environment, in a normal condition, okay? And so that's what your control group is. It's your normal condition. And we use this to compare. Otherwise, how would we really know if that independent variable is making any difference? Okay? Looking at this chart right here, this chart is just an example of how you would collect data, because remember we're thinking about what types of data can be collected. In here, they're showing you that there's a wet and dry environment for your isopods, and you're just counting how many are there, and you're also taking some other notes. So two types of data that you can use in an experiment, or that are usually collected when performing an experiment, are qualitative and quantitative data. So qualitative, quality, this can be kind of subjective, right? So qualitative data could be something like the texture, thinking back to your diffusion lab, the texture of the egg or the potato could have changed. It could be something like color, okay? Fish can change color, their scales can change color depending on their health, and that could be a quality type of thing, because that's sometimes very hard to measure. And we are going to be looking at behavior, all right? Such as crawling, okay, crawling over each other, climbing around, maybe just being still, okay? Movement in general could be a behavioral type form of data, or qualitative form of data. Where, on the other hand, we have quantitative data, and quantitative, just like quantity, of course, applies to numbers, okay? So in qualitative data, we want to make measurements, or we want to take measurements, such as things of continuous length. So that could be like a plant growing over time. We can also...
Francesco Sanchio, please come to the front office. Francesco Sanchio, please come to the front office.
We can also collect counting data, so whole numbers, how many isopods are there. But the one that's the most useful to scientists are measurements that are continuous, okay? And that's because these can be analyzed deeper. At the quote at the very, very bottom of this section says, what does this really mean? Does this represent useful or meaningful results, and how could I test this? And when you think about what you can do, the biggest thing that you can do to help ensure that your data is good is that you repeat it. Okay? The more times you can repeat your experiment and get the same results, the more reliable your results or your data becomes. So how do you determine the meaning of the data?
Francesco Sangio, please come back to the front office. Francesco Sangio, please come back to the front office.
You can determine the meaning by a statistical analysis of measurement data. So, when we're thinking of measurement, again, this is going to be not your counting data, not counting data, but you want that continuous data. That's going to be something that you can make more meaning from. When you're using statistics, you're actually determining the significance. That looks a little funny. I don't know if I spelled that wrong or not. But moving on. The distribution of your data, the layout of your data, you can compare it, or it can be separated as causation versus correlation. Okay? Causation. When this is going to be, and the question you want to ask yourself if this is a causation type of data is, did your independent variable cause a change, that's the delta sign of the triangle, in the dependent variable? Okay? So in causation, did independent cause the change in the dependent variable? Whereas in correlation, there might have been some sort of change, but the change was caused by some other underlying factor. It wasn't something very specific that we were searching for. So the relationship here is not caused, and there are those other factors available. We're going to quickly move on to our hypothesis. So with hypothesis testing, in your traditional type of hypothesis, you have what is called your if-then statement. And your hypothesis is usually set up as if ... the independent variable does whatever, then ... the dependent variable reacts in a certain way ... because ... Okay? This is your traditional type of hypothesis. On the other hand, you have another type of hypothesis called a null hypothesis, HO, okay? And that's a subscript. In a null hypothesis, what that means is that there is no significant difference between the conditions. Okay? And you can use data to accept or reject the null hypothesis, HO, but you cannot prove but can support that there is a difference, but the data will support that the null hypothesis And that's the big thing there, is that you have to remember that we can never prove something, but what we can do is we can say that the null hypothesis is not true, meaning there is some sort of significance in our condition.